So welcome back from the break. Hopefully you took some time out and now you're ready to hear the answer to these questions. So why are prokaryotes successful? Why are they so adaptable? Well, part of it is because they can exchange genes that allow them to adapt. So if one species of bacterium has genes that allow it to adapt to an environment, you can share those genes around. And so various bacteria can actually accumulate various adaptations that have occurred in different species and accumulate a combination of those adaptations to actually take on a better or be more successful, make better use of their environment. In fact, we think that's how aerobic respiration evolved in bacteria, by accumulating a series of mutations from different species that accumulated into a single species of bacteria that became the precursor of all aerobic bacteria. Another adaptation is their ability to reproduce quickly. So if something has if a species of bacteria has an adaptation that allows them to survive and thrive in a particular environment, they can grow to very large numbers very quickly because they're reproducing asexually, and they do so very rapidly. Remember, 1 to 3 hours average generation time between divisions. Of course, they can tolerate harsh environments that other organisms simply could not tolerate. They have that chemical adaptability. They have cell walls around the outside that protect them. They have capsules around the outside that protect them. They have all sorts of structural features that allow them to utilize the resources that they encounter. So that's a reason why they are so successful, or reasons why they're so successful. How do the cellular and DNA structures of prokaryotes differ from eukaryotes? So remember, there's no nucleus. So the DNA is in a nucleoid region. It's in a specific region, but it's not defined by the presence of a nucleus. The DNA is in the form of a large circular DNA molecule that represents the genome, plus small circular DNA molecules, the plasmids, that represent the transferable DNA. Obviously in eukaryotes, the situation is the linear DNA molecules and multiple linear DNA molecules of different sizes. We refer to those, of course, as the chromosomes. Then, of course, we have a capsule around the outside. We have flagella that allow for locomotion. We have sensory apparatus that allows the cells to sense their environment and respond to it. Remember, the flagella of prokaryotes are very different in form to the flagella of eukaryotes. If you're not sure what the flagella of eukaryotes look like, you might want to go back and take a look at chapter 6 in the textbook, in the Bio 1 textbook, where you'll find a description of eukaryotic flagella, the so-called 9+2 microtubule structure. Very, very different. So there's some features that differentiate prokaryotes from eukaryotes, and you can probably come up with a whole lot more as well. And you perhaps should try and make a list comparing the two. Anyway, onwards and upwards, let's continue on. So another feature that makes prokaryotes really successful is the fact that they have a lot of genetic variation. Now, you might think, well, they only reproduce asexually. They don't reproduce sexually. And we know that sexual reproduction from previous discussions is critical for maintaining genetic variation. So how do prokaryotes get genetic variation? Well, remember, they do have the ability to reproduce very rapidly. So once a particular prokaryote develops an advantageous mutation, it can get transmitted to a large number of individuals in the population very quickly, simply by asexual reproduction. Then they have the only source of internal new genes is through mutation, but we have to remember that mutation occurs spontaneously. And while not a lot of mutations turn out to be beneficial, those that do turn out to be beneficial are going to be transmitted into the population really quickly because of the rapid reproduction. But then we also have to factor in that bacteria practice 3 different forms of genetic recombination, transferring DNA from one bacterial cell to another. And it doesn't necessarily have to be a bacterial cell of the same species. It can be bacteria of different species. So all of these allow bacteria to create a wide variety of genetic diversity and genetic variation, even though they don't practice sexual reproduction, which is what we typically point to as the primary source of genetic variation in eukaryotes. So let's take a look at these. So we do know that they reproduce quickly. We've already talked about that previously, and it's by binary fission, a form of asexual reproduction. And that does mean that they can transfer favorable mutations into the population really quickly. So that's a source of genetic variation, but it's this, what's referred to as horizontal gene transfer, that really gives prokaryotes their advantage genetically. And one of the things that has been done, this is a very, very long-term experiment. So you don't really get an idea of just how long this experiment continued for. Oops, that's not what I want to do. I'm going to have to figure out how I can get this back in here now. There we go. That's good. Make sure the pen's working. Check it back to red. So if you go into the textbook and actually look at the text box where this is talked about, they tell you that this took place over 3,000 days. So it took 3,000 days to get 20,000 generations. Well, 3,000 days is about 8 years, and it was about 0.3 of a year, so that would be about 8 years and 4 months that this experiment took place. And it was basically this. They started out with a single test tube that contained 9.9 mils of broth that was suitable for E. coli to grow in. They added 0.1 mil of an existing E. coli solution into that test tube. The next day, they took a new test tube, placed 9.9 mils of broth in there, took 0.1 mil from the culture tube from the previous day and added that to this new tube, and they'll let it grow. On day 3, they did the same thing. And so for 3,000 days, they did that. They didn't do any manipulations other than transferring from one test tube to the next test tube on a daily basis. All they did then was monitor what was happening to the bacteria. Periodically, they would take a sample and take a look at what genetic changes were occurring. One of the genetic changes was real easy to observe, the rate at which the bacteria were growing. They could show that over the course of time, the rate at which the bacteria regenerated increased over time. So clearly mutations were occurring that were generating bacteria that could replicate much faster in these ideal conditions. But they also noticed that spontaneously antibiotic resistance mutations turned up, other mutations in metabolic pathways turned up with no selective pressure. These were just spontaneous mutations that turned up in these bacteria just because they were replicating and being allowed to replicate in conditions that were favorable to their growth. That was it. There was no evolutionary pressure applied to them. And so simply because bacteria have what you might refer to as a haploid genome. Let me just put this in here. Bacteria have a haploid. And I'll put it in quotes because, while technically correct, it is to say that it's haploid. There's a little argument backwards and forwards about whether it's appropriate to use that term. A haploid genome. So one copy of each gene. So that means mutations in genes almost always are going to generate something new, because there's no backup copy of the gene. When the gene gets mutated, you're going to create something new. If it's favorable to the cell, obviously that's an advantage, because that cell is now going to outgrow its competitors. And that advantageous mutation gets propagated into the broader population as those cells outgrow all the others without the advantageous mutation. So what you're seeing here in this growth curve is the accumulation of advantageous mutations and the displacement of those cells that don't have these advantageous mutations over time, simply because you're dealing with a haploid genome. Any mutation that is advantageous is going to have an effect, because there is no backup alternative copy of the gene. Every mutation is going to have an effect. So this is an interesting experiment. Obviously, it required some dedication to doing the experiment. The paper was published in 2000. You can read about the experiment in the textbook in the text box. But it reinforces the fact again that genetic variation is going to occur in a relatively short amount of time in prokaryotes, because of the advantage that they have of a haploid genome, rapid replication, and the ability, as you'll see in a moment, to transfer those advantageous mutations to other bacteria. So what type of genetic recombination do we have here? You can see the names of the three here: transformation, transduction, and conjugation. You may have already encountered transformation before, because this was discovered, and now I'm going to have a senior moment and forget the name of the physician who did this. Hopefully you learnt about this in Bio 1, because transformation was discovered as a means for changing the phenotype and the underlying genotype of bacteria back in the 1920s and 1930s. And it was the recognition that transformation that could occur that should have been an indication to the biologists of the day that DNA was a potential candidate as the molecule of inheritance, even though people were thinking there was probably protein at the time. So transformation is all about transferring existing DNA from one bacterial strain to another bacterial strain. This is the uptake of DNA in the laboratory. It's the uptake of plasmids. In the environment, it can be the uptake of plasmids, or it could just be the uptake of loose pieces of genomic DNA that have, when a bacterium dies, its DNA can become fragmented, and other bacteria can take up some of those fragments by transformation. So transformation is the uptake of DNA from the environment. Transduction uses viruses, or specifically bacteriophages, as vectors to transfer bacterial DNA from one cell to another, and I'll show you how that works. And then conjugation is probably as close as you're ever going to get to bacterial sex. This is where you've got direct DNA transfer from one bacterial cell to another via a physical connection between the two cells. So let's take a look at these three forms of DNA transfer. So as I said, transformation is simply picking up foreign DNA from the environment. Pretty straightforward process. Transduction involves the use of bacteriophages. Now, viruses, hopefully you understand how a virus works, but if you don't, a virus inserts its own DNA into the cell that it wants to infect. And viruses infect all sorts of cells. So there are viruses that specialize in infecting animal cells. There are viruses that specialize in infecting plant cells. There are viruses that specialize in infecting fungi. There are viruses that specialize in infecting bacteria. Those that infect bacteria are called bacteriophages. There are DNA viruses and there are RNA viruses. Here we're looking at a DNA virus. Now, under normal circumstances, the virus inserts its own DNA into the bacterial cell, and that triggers the bacterial cell then to become a virus factory. And what it does is start to generate the virus proteins and the virus DNA to create more virus particles. And most of the time, the new virus particles encapsulate viral DNA so that this becomes an infectious agent to infect other bacterial cells. But occasionally, a virus particle will mispackage not viral DNA, but host bacterial DNA. The virus particle will attach itself to another bacterial cell and will insert that DNA. But because it is not viral DNA, it's not creating a viral infection and turning the new cell into a virus factory. Instead, it inserts the bacterial DNA, which then gets incorporated into the genome of the recipient bacterium, and it becomes a recombinant cell because its genome is now a combination or a recombination of the existing genome plus the bacterial DNA from the donor cell. I have actually utilized this technique in the micro lab that I was working in to try and create mutant bacteria by working on the basic assumption that a percentage of the virus particles that I was growing would contain bacterial DNA. I would carry out methodologies that would potentially mutate that DNA inside the virus particles. And then I would use those hypothetically mutated virus particles to insert their bacterial DNA into my recipient cells. It was a long shot experiment. Did I get viral transmission of DNA? Yes, but I didn't get the production of mutated cells. When we analyzed the experiments, we drew two conclusions. Number one, that perhaps the techniques that we were using to try and mutate the bacterial DNA in the virus particles simply did not give rise to mutations. Or if it did give rise to mutations, it wasn't giving rise to mutations in the bacterial system that we were looking at. Because again, we were looking at a particular form of taxus, aerotaxis, and we wanted to try and mutate that system. And it turned out that we were unable to do it by trying to mutate bacterial DNA in virus particles. But the technique works. I can assure you this technique works. It works in the laboratory circumstance. And so obviously it does work in nature as well. So it is another way to transfer bacterial genes from one bacterium to another. And because bacteriophages can infect bacteria of different strains and different species. And we'll talk about what's meant by strain versus species when we talk about bacterial phylogeny a little bit later. Because bacteria can, because the virus or the bacteriophages can infect across strains and species of bacteria, you get this potential for horizontal gene transfer between different species of bacteria as a consequence of this. Then we have the notion of conjugation. Now, conjugation does not occur arbitrarily. It is driven by a series of genes inserted into a piece of DNA called the F factor. The genes in the F factor trigger the formation of a physical connection, an extension of the plasma membrane of the donor cell that fuses to the plasma membrane of the recipient cell and forms a hollow membrane-bound tube that connects the two cells together. This is referred to as a sex pili. So here you can see why I say this is probably as close as you can get to bacterial sex, because we actually use the term sex pili to refer to this physical connection. The location of the F factor determines what gets transferred in as DNA in terms of DNA from one cell to the other. So there is something called an F plasmid that contains the F factor. And if that is the case, then the F plasmid itself is what gets transferred. And that can be used in a genetic engineering context, because like any plasmid, the F plasmid can have genes inserted into it. And you can use that as a way of transferring genes from one bacterium to another by taking advantage of the formation of the sex pilus and the transfer of the F plasmid via the F pilus, along with the genes that you've inserted into the plasmid. Of course, doing transformation in the laboratory is a much neater way of doing it and probably a much simpler way of doing it, but the possibility exists being able to do that. If the F factor occurs in the bacterial genome, then you're going to get genomic transfer. So let's take a look at some pictures of the two processes. So here you can see the sex pilus linking these two bacteria together, and you can tell these are of distinctly different species. This one has fimbriae all over the surface of it. This one lacks those. So these are bacteria of two separate species, or at least two separate strains, and they're about to exchange DNA as a consequence of this connection. So if it's in the plasmid, you get the formation of the pilus. The F plus cell is the cell that contains the F plasmid with its F factor, and you get the transfer of the F plasmid across into the cell, the recipient cell, which is officially designated F minus for the time being. Once the transfer has occurred, the pilus breaks, and now you have two cells, each containing the F plasmid. So this cell has been converted from an F minus to an F plus by picking up the F plasmid from the donor cell. And again, if you have inserted genes into an F plasmid, you can have those transferred across as the F plasmid is transferred across. Not the most efficient way of transferring or of getting genes into a bacterial cell, but at least it's a way of doing it if you want. If it's on the if the F factor is in the genome, now you have the potential for genomic transfer, and now you get horizontal gene transfer because you're actually transferring genomic DNA between two bacteria. So here you see the F factor sitting in the genome of the donor cell. Here is our F minus cell. The transfer has to be to an F minus cell, because if it's an F plus cell, both cells are going to want to form the F plasmid and transfer DNA. So the transfer is always from an F plus to an F minus cell. I should have pointed that out earlier. So the pilus forms in response to the F factor activity, and now you get a transfer of genomic DNA from one cell to the other. That genomic DNA can then be incorporated into the genome of the recipient cell if the DNA is not degraded by protective enzymes. So there are different types of protective enzymes to protect against the insertion of DNA, of foreign DNA or foreign obtained DNA into the bacterial genome. One that has a wide use in genetic engineering techniques is a group of enzymes called restriction enzymes. These are enzymes that cut DNA, and they are bacterially derived, and many species of bacteria make their own unique type of restriction enzyme. But what is significant about these is they recognize specific sequences of nucleotides, usually somewhere around 6 to 10 nucleotides long. They may be reciprocal sequences. So if you read them from left to right or right to left, they read the same on each of the two. So one strand will read a particular sequence of nucleotides left to right. The other strand will read the same sequence of nucleotides right to left. In other words, they both read the same sequence of nucleotides in probably the 3' to 5' or the 5' to 3', depending on which direction you're reading the two strands. But they are reciprocal. They are symmetrical as a consequence. Others are not symmetrical, but they do recognize these unique sequences of nucleotides, and that's where they cut the DNA. Why these act as protective mechanisms is because the host bacterium does not contain those sequences within its own genome. So the enzyme is only ever going to cut DNA derived from an outside source. But it's going to cut the DNA up into fragments. And some of those fragments may actually get taken up into the bacterial genome. So it's not going to be a complete uptake of the donor bacterium's genome. It's going to be fragments of the genome. And not all the fragments are going to be picked up. But it does create a recombinant cell. The F factor is never incorporated. So you're not going to change the recipient bacterium from F- to F+. But it does pick up sequences that are derived from the donor bacterium. So this is another way to transfer genomic DNA. So you create a recombinant bacterium that contains genomic DNA. This has been particularly important in archaea, but we know bacteria do this as well in terms of gene exchange. This is horizontal gene transfer. This is very common amongst prokaryotes of all descriptions. Then we have the R plasmids. So the R plasmids are special plasmids that carry the antibiotic resistance genes. Most of the antibiotic resistance genes seem to be carried on these R plasmids. And remember again, plasmids are easily transferred from one bacterium to another, whether they be within the cells, whether they be released into the environment and picked up from the environment. Plasmids are easily picked up by bacteria. And they don't have to be passed from bacteria to bacteria of the same strain. They can be passed to bacteria of different strains in different species. So this is why antibiotic resistance is such a problem, because of this ability to transfer these plasmids. So let's take another break. Here's the concept check question. Shouldn't be a difficult one. Briefly describe the 3 mechanisms by which bacteria can transfer DNA between bacterial cells. See you on the other side of the break with the answer.